Hi, my name is Samuel Borges Cardona. I study in third semester of medicine in the Universidad Pontificia Bolivariana, and today we're going to talk about the selection and identification of an RNA aptamer that specifically binds the HIV-1 capsid lattice and inhibits viral replication. So, first we're going to talk about HIV. HIV is the human immunodeficiency virus that attacks the immune system and attacks the immune system. If it doesn't get treated, it can lead to AIDS, that is the acquired immune deficiency syndrome that can cause death. So, with the proper treatment, someone that has HIV can live a normal life and protect himself and his partners. So, all you have to do if you have HIV, no matter if you had it for life, you have to treat yourself. HIV is classified as a retrovirus that belongs to the lentivirus family. What is the lentivirus? The lentivirus is basically an exogenous retrovirus that cause persistent infections leading to multiple diseases that take long periods of incubation. Then we have the HIV replication. The HIV replication is basically the HIV gets to the surface of the host cell, joins with it, and introduces his RNA and some other proteins for it to create the viral DNA. It introduces RNA and proteins to the cell, creates viral DNA, and joins it with the cell DNA. Then to produce more new viral RNA and more immature HIV that later will come out as mature HIV to continue the replication process again. So the objective of the study that we're gonna explain today is to demonstrate that aptamer CA15-2 significantly inhibits HIV-1 replication in producer cells but not in target cells. And in the study we use these methods. The PCR. The PCR is based on the enzymatic amplification of genetic material. In this study, it was used to generate transcription templates amplifying the expression of plasmids in the aptamer used in the experiment. We also use genetic recombination. It refers to the rearrangement of DNA sequences by the breakage and rejoining of one to the another. And in this study was used to select RNA aptamers with affinity to the assembled HIV-1 CA lattice. We also use the page. The page method is a technique that we use to separate proteins and molecules according to electrophoretic mobility. In this one, we use the SDS page that we use to confirm the purity of the aptamers. And we also use EMSA, that is the one we use more because we have to evaluate a lot of properties of the CA15-2 aptamer. So, the results of this we have here an EMSA. This is an EMSA, and this is 1 and 2. This EMSA demonstrates formation of a complex between the aptamer CA15-2 and the CA lattice. Aptamer CA15-2 exhibit a significant mobility shift in the presence of CA lattice, like we see here. And the next one is another EMSA to determine the ability of unlabeled competitors to compete with aptamer CM52 for binding to the assembled CA lattice. Unlabeled full-length CA52 was able to compete for binding to a greater degree than non-binding control RNAs, like we see here, and comparing to competitors. We also have another EMSA that it was used to determine the ability of unlabeled truncated aptamers to compete with aptamer CM15-2 for binding to the CA lattice. These results show the full-length aptamer, the CA15-2, and truncation of the same one, but 180, 170, and 160. We see that as increase the comparator concentration, it is decreased mobility shift in the binding to the CA lattice. What can we say about it? That the 160 generates a more stable structure capable of binding to the lattice than the others. Yes. And we have here the modified CA52 aptamer structure that we use in the study. Then we have the discussion. Three authors made a statement of related to the study, and these are the ones. So, Sejutina said that due to the cross-linking, these stable tubes can be removed from high salt buffers without disrupting the lattice and have been shown to maintain interactions with lattice binding host factors, which agrees with this study. 10 said that the immature lattice is formed via recruitment of gag dimers to the site of assembly to form aptamers, which then assemble into the immature lattice, which also agrees with the study. And Lange said that we have previously demonstrated that at the discussion of aptamer expression, plasmid utilizing these experiments, aptamers, including non-binary controls, can be non-specifically encapsulated into viral particles, which also agrees with the study. And with conclusions of the study, we have that experimenting with the structure of HIV virus using different biological techniques help us to know its functioning and how can we apply knowledge to create ways to control the disease more. And understanding how the replication of the HIV structure, we can learn how to stop it, and this way manage to make a step forward in the fight against the disease that has been affecting this world for so long. Thank you.